Welcome, everybody. Welcome to this third session of this webinar series on vigilance of COVID-19 vaccines hosted by the International Society of Pharmacovigilance, the ISoP. This webinar series has been organized by the Latin American chapter in collaboration with the ISoP Special Interest Group of Vaccines and the ISoP Israel chapter. My colleagues and co-chairs are Rebecca Chandler from the Vaccines Special Interest Group and Irene Fermon from the ISoP Israel chapter. My name is Raquel Herrera Comoglio. I'm from the National University of Córdoba, Argentina, and from the Latin American chapter. Today, we have the great honor to announce that the Professor Saad Shakir will speak about the observations and experience on vaccination of COVID-19. Saad Shakir is a pharmacoepidemiologist and drug safety physician. He has worked in the fields of pharmacovigilance, pharmacoepidemiology, and risk management for 30 years, initially at the UK Regulatory Authority, then the international pharmaceutical industry, and as a director of the Drug Safety Research Unit in Southampton. The Drug Safety Research Unit is an academic unit associated with the University of Portsmouth. At the DSRU, Saad leads a research team with an active program for monitoring and studying the safety of medicines in populations. He has led many important drug safety studies and has worked and advised on many drug safety issues, including product withdrawals, major restrictions, and important safety measures. Saad serves as a chairman or member of safety advisory boards and data safety management committees. He is an author of many book chapters and publications in scientific journals on pharmacovigilance, pharmacoepidemiology, and risk management, and is a member of the editorial boards for the journals Drug Safety and Pharmacoepidemiology and Drug Safety. Saad Shakir has led and coordinated many postgraduate educational and training programs, including the Master Science program in pharmacovigilance at the Drug Safety Research Unit. Professor Saad Shakir, please go ahead.
Okay. Well, good day, everybody. Can you hear me?
Yes.
Yes.
Okay. Well, I'm grateful to ISoP, the Latin American and Israeli chapters, and the ISoP Special Interest Group for inviting me. As Raquel said, this is sort of a broad presentation, focuses obviously on safety and pharmacovigilance about COVID-19 vaccines. As some of you know, I am involved with quite a number of studies. I'm now the principal investigator for 4 studies, one of them which we have just launched on the AstraZeneca vaccine, the study on the CPRD study on the Pfizer vaccine, and we are doing 2 other studies on all vaccines which will be used in the UK. So kind of heavily involved. So we'll start to say what happens conventionally, and this is from the FDA. I think many of you are familiar with the phase 1, phase 2, phase 3. It takes 10 to 12 years, and these phases, the first phases, well, in vaccines, all in healthy volunteers, but they started in phase 1 with up to 100 volunteers, then it goes several hundreds in phase 2, and then in phase 2 and phase 3, they said hundreds of thousands in the US, but in Europe, it has been said that it required 60,000 in the conventional sense of developing, in a conventional process of developing a vaccine. As you all know, this has been shrunk to 8 or 9 months, and we now have several vaccines. For the SARS-CoV-2 or the COVID, the regulators modified the process, as you know, with the rolling evaluations, and the pharmaceutical companies changed the development into parallel rather than sequential development. And the requirements have been that they have phase 3 trials for about 30,000 people, and some of them hit more than that, and others hit slightly less than that. I mean, it is really not known widely that all these vaccinations are interim authorization. They are not full authorizations, but still with that interim, we are getting millions and millions. And the idea is that we'll continue to learn. I mean, for the studies, actually, the randomization continues. I know for the AstraZeneca vaccine, the people who were randomized to take either the AstraZeneca vaccine or the comparators continue in the study until they are called to be vaccinated. At that stage, they will stop being involved in the studies, but the company had asked them to contact the company so that they get blood from them to do testing. So the studies are to continue, and the idea is that we will hear more results as the time progresses. Obviously, in countries where the vaccinations programs are progressing well, you will have more and more patients will be censored, and their observation will be sort of terminated. So this is it. I mean, the shrinking of the development, the shrinking of the numbers, and all the regulators said that like the licensing process was thorough, no corners were sort of turned inappropriately, and so on. But the fact remains that there was limited observation for safety and efficacy. I'll come to address this because there's a lot of people talk about there is no long-term observation and so on and so forth. And I'm going to address how important that is. Now, this is really one of the most successful endeavors not in the history of medicine and vaccination. I mean, this is the time between identifying the pathogen and developing a vaccine. And you see that in meningitis, it took 80 years. In whooping cough, it took from sort of the 40 years, and so on and so forth. While we are having this is almost like a wonder of the modern world, that it took us really less than a year, in fact, about 3/4 of a year, to come up with the vaccine. And you know, the COVID or the virus was opportunistic, and it came to us through whatever means it came to mankind. But it couldn't, you know, it is sort of, it came at a time where we are in the middle of the genetic revolutions. And that's why it was possible to utilize the genetic revolutions. As you know, the identification of the genotype of the virus, you know, happened, you know, about 2 months of identification of the virus. And this is something which I think people don't recognize how important and how marvelous this was, although it's obviously a short RNA, but the ability of the science to not only to identify the sequence, but to develop vaccines for the sequence is a wonder of science. Now, there are, as you know, all sorts of mechanisms for the vaccines. So there's the whole virus inactivated vaccines, they're part of the proteins of the virus. There's the DNA virus. I'm going to kind of focus on two vaccines, the vaccine which uses the lipid nanoparticle and like Mrna and Pfizer, and the vaccines which use the viral vectors like AstraZeneca and Sputnik V. Just to take you in the background, to get genetic material inside the cell has been an active area of research for more than 30 years. How do we get genetic material inside the cell, whether it's RNA or DNA? And basically, and that is really in all genetic therapies, not only for vaccines, there are two broad approaches to do that. Either you put the genetic material inside a lipid nanoparticle, or you put it inside the virus, and these are the two approaches where you get genetic material into the human cells. And here are the RNA sort of in the envelope in the nanoparticles like the Mrna and the virus. They go to the cell wall, which is a lipoprotein, and then they diffuse, and the RNA will interact with the cellular RNA and start to produce the surface antigen, which everybody in the world recognizes. And then the surface antigen will go to the cell wall, and then antibodies will come to that surface antigen, and also T lymphocytes will come to it. That is, I think, the process which everybody knows about it. With lipid nanoparticle, all that happens inside what is called the cytoplasmic soup. And this is a point which I'll come to again. It doesn't go inside the nucleus of the cell, and this RNA, which is sort of enters the cell, will be degraded, and will be degraded over a short period of time. While when the RNA is part of a DNA virus, like the virus which they have been using, the adenovirus of the chimpanzee, where genetically it has been modified previously, where 20% of its genetic material was modified, and it was used for Ebola virus, and then the RNA is incorporated into the DNA of the cell, and when the DNA of the virus, and when the virus enters, it goes inside the nucleus of the cell, and it starts the process of producing the antigen. There's 2 points to make here, because a lot of comments about these vaccines are being made. Neither the vehicle, when it is a nanoparticle, nor when it is an adenovirus, actually becomes part of the human genetic material, whether it is RNA and DNA. That is a fact. It doesn't become part of the human DNA or RNA. This is one fact. The other fact that the virus is non-replicating. It is designed not to replicate. So it goes there, it produces the antigen, then it dies and it disintegrates. So in fact, if you ask us, we want them to stay a little bit longer. We want the RNA, or we want both inside and outside the nucleus. We actually want it to stay a little bit longer so that it can produce more antigen for us, so that it can sort of stimulate more antibody production. But the fact is that they don't stay, and they don't interact with the human DNA. And that's another pictorial way of it. Now, that is the mechanism. Now, as I said, the regulators said that, for the COVID vaccines, they needed 30,000-ish patients in the phase 3 studies. They also required other things. They required that 65 people above the age of 65 years should represent at least a quarter of the total number of the participants in the studies, because as we all know, age is a risk factor, and these are the people who are at the highest risk of getting severe COVID and fatal COVID. And most of the studies have this number. As you know, the AstraZeneca vaccine, because of all sorts of complications during development, didn't have 25%. In fact, the AstraZeneca program had about 10%. And that's what has caused different decisions by different regulators. The UK and the EMEA, or the UK starts first, they accepted the fact that this number is smaller than 25%, but they looked at the immunogenicity of that vaccine in the phase 2 studies. And the immunogenicity, i.e. production of antibodies, was as high above the age of 65 as it was in the younger age population. And they accepted looking at the science in a broad way and saying that they didn't think that there will be differences in the ability of the virus to offer protection, of the vaccine to offer protection for the people the age of 65. The EMEA, EMA has accepted that, the WHO accepted that, and we know some countries like Germany, France, and Sweden, and others didn't think that was acceptable, and they are waiting to see more results from patients above the 65. The company has 2 studies, and also, as I said, there is a rolling evaluation, and more people were included in the studies later. So this data will become known. Also, we now have a significant number of, for example, people in the UK, out of the 16 million plus who are vaccinated, a significant number of them, quite a large number of them, are above the age of 65. So we will get the results of both real-world results of both their antigenicity and also whether they will be protected from that vaccine. Another area which the regulators wanted significant numbers in the trials were actually people from ethnic minorities. We have been talking about different genetic responses to medicines and vaccines, and these were required to be presented in the cohorts. Now, this is an issue which I want to take. In the UK, as we all know, they decided that the MHRA, our regulatory authority, the Joint Committee of Vaccinations, and the chief medical officers to expand the period between the 2 jabs from the 3 and 4 weeks, depending on trials, to the 4 to 12 weeks, but nearer to 12 weeks. And the argument was that both the efficacy of the Oxford AstraZeneca and the Pfizer vaccines before the second dose were 73 and 88% respectively before the second formal dose, 3 weeks and 4 weeks. And the understanding or the assumption or that will not decline over the 12-week period. And they did not only the results, as they did look at not only at results from the final interim reports from these studies, but because of the AstraZeneca vaccine was the second dose, because of production difficulties during the clinical trials, were given between periods of 4 weeks up to 26 weeks, up to 6 months. They looked at what's called exploratory analysis, and they found that the number, the best efficacy was provided on these exploratory analysis was provided at 12 weeks. So that's why they concluded that 12 weeks is the best period. It is sort of a pragmatic decision on a smallish number of patients based on what's called exploratory analysis. And also the decision was not only looking at the trials and the vaccines, they looked at what was a very steep rise in the incidence of COVID cases in the UK, and they felt that they needed to do something about that. The hospital ITUs were full, the deaths were very high, and they felt that a combination of a lockdown and introducing the vaccine to the highest vulnerable people was the right decision. And there was an assertion that the benefit of the first dose over the short-term protection, while the second dose, not only for COVID vaccines, but in vaccines in general, are to provide a longer-term protection. So the first dose will give you the protection, the short-term protection, and short-term is not like in days and weeks, the short-term, months, but the second dose in vaccines provide the long-term protection, and I will come with an example. And the other efficacy of other vaccines, hepatitis and so on, show that if you give the first dose, you'll get a shorter period of protection, but not less protection than when you increase the doses. Now, we had or the UK or AstraZeneca had a lot of criticisms from scientifically trained people, experts, but also from gentlemen like Monsieur Emmanuel Macron. Now, after about last week, the WHO recommended that the Oxford AstraZeneca COVID vaccine, it recommended that it can be given for 12 weeks between the two doses. And I am looking at interest to see, because of the issue which we're going to talk about, because of the difficulty in getting vaccines fast in some countries in Europe, whether some of them will follow the UK and the WHO decisions to recommend 12 jabs. I mean, in the UK now we have 16 million people. Many of them received 1 dose. We need to do modeling to compare whether the protection from that model is better or worse than if we gave the 2 doses within the 3 and 4 weeks. So we compare 16 million mostly with 1 dose, or 8 million or 8.5 million with 2 doses as per recommended initially, and what would be the impact of that on public health. I'm very, very sorry I don't have the slide, but the decline in infections and hospitalizations and slightly early in deaths among the above 80 years old is going down faster than other age group because they will be vaccinated early with the first dose. So although the lockdown applies to all age groups, the vaccination applies to the above 80s, and now we are seeing real world data supporting that vaccinating the largest number of the vulnerable had a protective effect. Now, this is, I'm not going to bore you with that, but this is from a hepatitis A inactivated vaccine. And that is the summary of product characteristics as approved by the EU. I know you can't sort of extrapolate, but this is for an inactivated hepatitis A. And it says in the ISoP there, the first dose is for the initial protection, and the second dose, which can be given somewhat flexible time after that, is for long-term protection. Initial protection is achieved with one single dose, and protection levels of the antibody may not be reached after 14 days of protection. So this was for, and in fact, the second dose for the hepatitis A is given by, between 16 to 12 months after the initial dose. So I was vaccinated with hepatitis A. I was told if I take one dose, it will protect me for 3 years. If I take the 2 doses within the period of between 6 and 12 months, it will protect me for 12 years. I mean, I'll be interested to hear what Rebecca would say, because what is the biological basis for choosing 3 or 4 weeks between these 2 vaccinations? Is there a biological basis for it, or is it for the convenience of compliance and the patients coming back? I fully understand the argument to say that's how the clinical trials were done. That is how the efficacy was shown, and you must stick to it. It's not a bad argument, and I understand it, but now we are testing the 12 weeks period in real world data. And we have the example of Israel, which chose to give the vaccinations according to the recommendations. So there is a lot for us, people working on on real world data, to think about. Now we go back to safety. As we know, risk management plan for all medicines and vaccines, the pillars for these are important potential risk, important identified risk, and missing information. And also we know that the benefit risk evaluation is a continuum between pre-marketing clinical trials and post-marketing. These are sort of the pillars of our understanding for risk with medicines and vaccines in general. And this is the risk management which we have been working on since the sort of the guidelines in 2005, which became regulations in ICH guidelines in 2005, which became regulations in the EU in 2012. So this is our paradigm. Now, these are, I apologize, it's a busy slide, but it's important to take us through each of these vaccines, because for the Pfizer vaccine, the important identified risk was anaphylaxis, and at the time of licensing, that was not a case with the AstraZeneca vaccine. The important potential vaccines is vaccine-associated enhanced risks, which is including vaccine-associated enhanced respiratory distress, and that is for all of them. And the regulators are interested in that, because with other vaccines, conventional vaccines, there have been cases where if patients break through the vaccine and develop an event, these events can be more severe form of the illness. I am very interested to know this, because here we're not giving the whole virus. We're just giving a small part, a very small part of the virus, which is part of its RNA. Whether that will be able to cause a vaccine-associated enhanced disease or not is something which I'm very eager to see, because I have difficulty in understanding how that may happen. There could be a surge in the antibodies because the antibody response and so on, but it will be interesting. That could happen. With the AstraZeneca vaccine, there is an additional thing, nervous system disorders, including immune-mediated in neurological conditions. And these came from the 2 cases of transverse myelitis, which have been very well publicized during clinical trials, and also some cases of facial nerve palsy, I think about 4. Now, anaphylaxis was included in the potential risk as opposed to the identified risk with AstraZeneca vaccine. All of them had missing information, pregnancy and breastfeeding, and that is a subject which I think all of us can talk about if you want to. Immunocompromised patients, we don't know. This is missing information. If you are immunocompromised, whether biologically because of a disease or because of a drug, will you mount a response? And to being vaccinated, and there is a lot of discussions about how severely or how aggressively you are immunocompromised. And there's a big question about corticosteroids. What amount of corticosteroids make you immunocompromised? There's a discussion about prednisolone 10 milligram being the level. Again, that's arbitrary. So this is something which we want to hear or want to learn from post-authorization study. Then came in the use and frail people with frail conditions, such as chronic obstructive pulmonary disease, diabetes, chronic neurological disease, and so on and so forth. That is missing information because we are looking for, and in observational studies, excuse me, which we and others are doing, we're trying to include as many people with these concurrent conditions to see whether the effect of the vaccine will be more in them. And I'm going to talk about the Norwegian experience later. Used in patients with autoimmune disease, all of them, this is missing information. We don't know what happens to these people. Long-term safety data. Because we have limited, I mean, the first person vaccinated back in May. So all what we have is 8 or 9 months. This is which vaxxers and the people are talking about. As Jean-Michel said last week, the thing is that we look at things which may happen in the future on the basis of evidence-based thinking, pharmacological thinking, vaccinological thinking. We don't think about, oh, something may happen. We want to look at long-term effect the base of some scientific rationale about them. I could be hit my house hit by lightning in the next hour, but there is no clouds above me. So I can't say that I will be hit by lightning when there is clear. I have to have evidence. I have to have some dark skies on above my house to say that my house may with lightning would like, and so what is likely? As I was saying about the cloud, I mean, okay, a lightning could hit my house, but when the sky is clear, there's no likelihood. But if there's cloud of my house, there is a risk that this lightning might hit my house. So we have to need evidence about these possible long-term effects. Interactions with other vaccines, they want to, that has been a concern. And we are asking whether people have had the flu vaccine within month before vaccination. And all of them, the risk management plan in Europe requires post-authorization safety studies of various kinds, whether it's a secondary data collection or primary data collections. Length of the studies, the original clinical trials were supposed to be for 2 years. I'm going to ask every time, can you still hear me? Can you still hear me?
Yes.
Yes. Okay, because I'm now panicking every minute. Right, okay. The studies were designed, the clinical trials, for 2 years, but people are being censored because they will get vaccinated. So observational studies will be important to look at long-term effects. This is the scheme which I had a few months ago, that we need to enhance spontaneous reporting, and spontaneous reporting has been enhanced. Many regulators have put special forms for reporting COVID medicines and COVID vaccines adverse events. We need active surveillance where we identify people and we follow them up. And we need secondary data sources. We have a lot of secondary data sources around the world, in the United States, Scandinavia, Europe, Germany, Netherlands, UK, CPRD, Spain, and all of them are being used to monitor COVID vaccines. We need add-on studies to do secular trend, case control studies, self-controlled case series. I'm not going to go into the details. And we need some biological studies sometimes if there are special issues about special subgroups. And there have been a couple, 2 or 3 cases of immune thrombocytopenia. We need to see why these people are getting their events. Some scientists may come and want to see what is the cause of that. This is an important point about the efficacy of vaccines. Everybody in the world talks about this vaccine is 86%, this is 93%, 60%. I mean, what matters is all of them in the studies, deaths from COVID-19, deaths from the vaccine, hospitalization from the vaccine, with all the vaccines that you see here, all these are 0. That there are no deaths from COVID, no deaths from the vaccine, and no hospitalizations in people who are vaccinated. And this, from protecting the people, I'm sure everybody agrees is important. Now there is the vaccine-enhanced disease. This has happened with other vaccines, and there is a concern about it. It has been seen in animal models with SARS and MERS, but it has not been demonstrated in any of the clinical trials and the vaccine studies. There is no vaccine-associated enhanced disease. We will monitor it for it. It is an adverse event of special interest by the regulators. Then there is adverse events following immunization. They are not necessarily AESI. They could happen as a result of anxiety, as a result of error, although there are checks of balances, as a result of the vaccine handling and also quality issues. We know that vaccines are complex biotechnology products. They are not like simple medicines where it is a chemical process. So many vaccine batches are thrown away and things like that. So we have, excuse me, to monitor for vaccine defects. That's why everybody in the vaccination card has the name of the vaccine and the batch number. So there are other, and this is the list of the AESI. I heard that Rebecca was talking about it. This has been developed by the WHO, Access, and others. And these are events which have been known associated with medicines, generalized convulsions, anaphylaxis, and so on. And the list continues, Guillain-Barré syndrome, narcolepsy, and so on. These are not necessarily things which have happened in clinical trials or will happen with COVID vaccines, but this is a list of adverse events which are known to be associated with vaccinations. And I'm not going to go through that. And here we have to compare the incidence of AESI. I'll just focus about these. We see what happens after COVID vaccines, but we have to know what is the background rate. So we compare the observed versus the expected, and the EU have funded a study to look at the incidence of these events of special interest in the underlying population who are not vaccinated, so that when these events happen, we will compare the observed with the expected. It's a very complex project because the measurements could be different using secondary data sources and so on, while the events will occur in primary data collection. But at least it gives us an indication of the kind of frequency which we expect. And now I'm going to talk about one of our studies which we are doing. It is to monitor the safety and utilization of the Aiza vaccine. Our objectives are to describe serious adverse events of special interest, and describe, characterize who, the demographics, who are getting it, other concurrent medications, other vaccines, and so on. And we study the population, people with immunodeficiency who are getting the vaccine, treat people who are in immunosuppressant. I may have mentioned that immunosuppressants is a long list. A lot of people in the society who are now on immunosuppressant. methotrexate for rheumatic conditions. And the question is, would steroids are immunosuppressant? And if so, at what level? People talk about prednisone 10 milligrams. So these are all things we are all learning. And so this is the study which we are doing. This is the business in Norway, where initially they reported in January 23 deaths of very old, not old, very old, and very frail, not frail. And these people really have a life expectancy in weeks, and 23 of them died initially out of 43 or 44 vaccinated people in Norway. And the question was, would this sort of the simple events which, well, not the simple, the non-serious events which occur soon after vaccination, the fever, the tachycardia, the diarrhea, and so on, which a normal person just can handle them, are these sufficient to sort of cause death in these very, very compromised elderly people? The Norwegian authority looked at these and the cases which came subsequently, 33 cases, and assessed causality to be unrelated to the vaccines, but didn't advise any change to the summary product characteristics, but advised that these people need doctors need to make a risk-benefit evaluation on these very old and very frail. And it's not easy, because these people are more likely to die from COVID. So the question is, if you vaccinated, would you make looking after them for the remaining days of their life better, that people are confident to go and nurse them, bring them the infection, their relatives can come and see them, and so on. So it's a very, very complex situation, and the advice is no change in the summary of product characteristics. And that is really the gist of it. Anti-vaccination is something which is giving us headache, because there is the anti-vaxxers. And this is a political, ideological movement. This is a 100-year-old slide which says forced to be vaccinated. And these people, we will not persuade them. If somebody thinks that Bill Gates is injecting microchips in their blood, we're not going to check. What we're concerned about is that they influence the vaccine hesitance. People are your neighbor, my neighbor, your colleague, who is saying, the vaccine have not been tested. Did they bring them too early? And they get influenced by these people. So our job is really to refute assertions made by these people. This is an example. Dr. Wolfgang Wodarg, a German doctor and epidemiologist, he asked the EMEA to delay approval of the virus vaccine because of concern about something called sinusitin-1, which is a protein component of the placenta. And he said that this was causing fertility hemoglobin, or might cause fertility, and he had a petition of a large number of people who signed his petition, and he submitted to the EMA. Obviously, EMA didn't think there is any evidence, and they didn't delay the licensing, the authorization. The Royal College of Gynecologists and Obstetricians in the UK issued a statement saying, we want to assure women that there is no evidence to suggest that COVID-19 will affect fertility. Claims of any effects of COVID-19 vaccine on fertility are speculative and are not supported by data. This has always been a job for us in pharmacovigilance, is to evaluate these claims and say what we think about them, usually to refute them. And we have to do that on the base. And that's why we, for example, at the DSRU, we produced a summary of the death of the elderly people in Norway. Yesterday, somebody died in Crete in Greece, a few minutes after being vaccinated. The Greek doctors said it was unrelated. A, it wasn't anaphylaxis. B, it was unrelated. The person just had a myocardial infarction. Myocardial infarctions occur, sudden deaths occur, and sometimes occur soon after vaccination. And anti-vaxxers will use them and will use other these. I mean, Wolfgang is a qualified doctor and so on, and he comes with these ideas. He is known to be an anti-vaxxer. Now, this is, I'm heading to the end. This is the problem with vaccine availability around the world. Can you hear me?
Yes, we hear. Yes, yes, we hear
Yeah.
you.
Yes, yes.
Okay. Okay. The population of the world is nearly 8 million. I think it's 8 billion. This is the number of vaccines which we have now for people, less than 4 billion. So as we stand now, we have less than 4 billion for a place of 4 billion for 8 billion. That's availability now. COVAX, which is, as you know, is an organization which was set by the WHO, France, and the EU to provide vaccines free to poor countries and at low cost to low-income country, and they committed to provide 2 billion doses in 2021. So the rest of the world will have 2 billion doses. I don't think COVAX will get that 2 billion. So we have vaccine shortages. And this is the capacity for producing vaccines by the various providers by the end of 21. So by the end of the 21, if all goes well, there will be 6 billion people potentially can be vaccinated. So we're still short of about 2 billion, and that may not happen. So there will be insufficient, and that will provide the vaccines for 2022 and later. So certainly by end of 2021, we're not going to vaccinate the majority of people in the world. And in 2022, that may not happen. And there is this competition between the rich country to get the vaccine. And it has been said, if no one is safe unless we are all safe. We are seeing all the vaccines. The world is not like 1918 with the Spanish flu. People travel, people move, people have relatives all over the world. So we really, if we want to control this pandemic, we have to work. The rich shouldn't sit down and say, I will vaccinate my people and put my head in an ostrich, because it will come to you. It will come to you. So we really, the world has a problem with providing vaccinations. Now, this is my last slide. It is an optimistic slide. I think in the whole history of medicine, not medicine, vaccines are second only to clean running water in saving human lives. I mean, and I put here to say, if we want a healthier and longer living humankind, if we provide these 5 things, clean running water, good diet, stop wars, vaccines, exercise, probably also, not probably, prevent infant and obstetric mortality. If we have these 6, we could close all the pharmacies, all the hospitals, all the surgical theater, and we can do better. We have no smallpox in the world. We are in the genetic era. We can do a lot. We have provided vaccines within few months. So I think, this is kind of my answer to the anti-vaxxers. These things are more important to the world than all the sophisticated method operations and medicines and so on we do. They're important. They're improving our quality of life. They are prolonging our lives, giving us good quality and so on. But these 5 or 6 things with infant and obstetric mortalities, and vaccine is one of them, are the sort of the pillars for providing a healthy sort of humankind. Thank you very much, and I'm sorry about the difficulties.
We have one question in the Q&A. How effective will these mRNA vaccine be against the mutated versions of the viruses?
I think, I mean, if in the meantime you want me to take a stab at that, I feel comfortable trying to do that.
Well, yes, I think in order to use the time,
Yeah.
I would suggest that if you can respond to this question, that's
Yeah.
why I'm asking.
So I'll answer this live. I mean, what I understand at the moment, I actually just came from a WHO working group meeting just prior to this, where we discussed the mutants, and the UK variant at the moment doesn't appear to be as much of a concern regarding the current vaccines that are being used. It's my understanding it's mostly the concern is about the South African and the Brazilian variants. But on the one positive thing about the mRNA vaccines, it's been my understanding that it will be this particular platform or technology is the most amenable to quickly being adjusted to adapt to any sort of variations that are ongoing. It's quite easy to get a new copy of the spike protein variant and to be able to use that piece in the vaccines. So my understanding is that this particular technology will be the most easily adaptable to any problems that we have with variants in the future.
So that means that it's preferable whenever possible to be vaccinated with an mRNA and vaccine. And you would assume that if we get the first shot with the one product which was not designed for a variant, and we get the second shot with the something with the variant, covering the variant, that would probably be effective? Or would you then, if someone has got the two shot with an mRNA vaccine, then you would recommend to give a third one, but maybe only one shot for the variant? What would be the
well, I think that, I must speak. This is just still a bit, I don't know how strong that we have, how strongly we can make these recommendations without hard evidence. I know that there's still some extrapolating going on, but I mean, the one issue with mRNA vaccines, although they can be easily adapted to new variants, this very much is a situation where you're only producing antibody to the spike protein itself. So the inactivated vaccines that we have, such as the Sinopharm vaccine, or even these viral vector vaccines, from my understanding, we actually get a much stronger T cell response in addition to the antibody response to the spike protein. So I think that there is some. Certainly in this WHO meeting where I was just now, we know that for the adenovirus vector vaccines, the neutralizing antibody titers are lower than with the mRNA vaccines, but we get a very strong T cell response. So I think that there are variations in the strength as well as the breadth of immune responses with these different platforms, and I don't know exactly how strong of conclusions we can draw at this point. I know that WHO has had some meetings even last week to discuss the impact of the arrival of these variants on the different vaccines that are out there. But I haven't heard anyone be able to make some strong recommendations yet.
Last week, we have discussed the idea of perhaps using several different vaccines sequentially so that we can provide different type of antibodies. And it was something that I think that UK has already started to speak about. So what is the position of WHO on this idea? I don't know. Is there some discussion going on this approach?
I will answer just that I think that there must be, but I haven't been a part of those. But I know that they have multiple different types of working groups ongoing. The one that I am actually a part of is actually to look at potential vaccine candidates for inclusion in their future solidarity trials. So we have a lot of discussions about immunology in particular But I know that there are accessory meetings that a lot of the members of this working group are also a part of. So I've just understood from their participation in the group I'm in that a lot of these discussions are ongoing in multiple different WHO circles, so to say.
There is also one ongoing discussion about pregnant women. We have seen different positions. The WHO has started to say that it's not advised, and then it has changed. Some association of gynecological obstetrics has advised to vaccinate. So it's really something that is moving a lot. Can you tell us about where we are today?
I feel that anyone who knows about any of these things is also free to let us know. I can tell you what I know about that. I do know that there is a move to start including pregnant women in some of these trials. So this has actually been discussed in the working group that I'm in. When they come, we hear vaccine candidates when they come with phase 1 and phase 2 data. And one of the criteria that we look at is whether or not they have any data in pregnant women. And my understanding is that the feeling on gathering that data in a clinical trial setting is very much ongoing and maybe starting actually. In terms of use, I can say with more assurance that at the UMC we've been working with the WHO Vaccine Safety Group to actually support their work that they have in their APHI surveillance recommendations in their WHO COVID-19 vaccine surveillance guide. So we're really working to adapt the tools that we have and provide in the WHO program of international drug monitoring. And one of the, we've just been informed that the APHI, the adverse event following immunization reporting sheet, for spontaneous reports or passive surveillance, those have been adapted to collect information in pregnant women in a more complete fashion. So with the thought that we know that given the mass vaccination campaigns that we will be vaccinating women who are pregnant, perhaps maybe even greater, that they're unknown that they're pregnant, but there certainly has been efforts ongoing to better collect that information in passive surveillance systems. And I'm sure that it's there in active surveillance systems as well.
In Israel, we have started to vaccinate pregnant women after several cases of very severe cases of COVID and even some deaths. I was very surprised because last week I had a question about if a woman has discovered that she's pregnant after the first jab. Is she able? Must she have the second jab? And it was a very difficult answer. And then I called an obstetrician. Friend of mine, and there is exactly this answer in the Ministry of Health recommendation that yes, she must receive the second jab. That I was very surprised that even in the first quarter they will receive this the second jab, and so it's moving all the time.
So just to move on to some questions and related, I see that Ulrich Hagemann has asked us about.
Yes, there are other questions. Yes, if you can answer the other questions. So
if there are any other theoretical considerations about the risk for the pregnant woman herself or the fetus newborn. And my answer to that is actually that I have not heard of any that are specifically vaccine platform related in any form. I mean, given the fact that the mRNA is short-lived and these viral vectors that we're talking about now are engineered to be non-replicating, I think from my work in vaccine safety that the only risks that I could think just might be related to the reactogenicity of these vaccines. I mean that there is a very high likelihood. We know a large percentage of people getting these vaccines do experience fever as primarily. And I mean, I have heard in my work, as well as when I was pregnant in myself, that there is sometimes thought to be a risk of high fevers in pregnant women and their potential effects on feti, I guess. But I mean, that most certainly has to be weighed against the potential risk of becoming severely ill and having a very strong immune response and poor outcome from the infection itself. So it's still something that I would think that in terms of fever, if that does play any role, that from a vaccine would be less of a concern than from the potential fallout from actually having the infection itself. I will move on to another, since we're talking about pregnant women and newborns. We also have a question about the fact that clinical trials are still yet to be done or just getting started in children. So should children also receive the current vaccines? And I think that this is a very difficult question. And I think the answers or the conclusions will be varied based upon, I think, cultural differences, honestly, between countries about these recommendations, cultural differences, maybe situations about schooling. I think it's a very complicated decision for a lot of countries at this point. Speaking for me personally, as a parent in Sweden, there is a plan here in Sweden to offer the vaccine to all persons greater than the age of 18 and to not include children. Most of you probably know that I believe Sweden might be one of the only countries in the world where our children have stayed in school throughout the entire pandemic. Our schools have never closed, in fact. And I think in Sweden, there has been a bit of soul searching and thinking about, if you recall, also in Sweden in the previous pandemic, the H1N1 pandemic in 2009, 2010. Sweden was one of the countries that noticed an increase in the cases of narcolepsy after receipt of the Pandemrix vaccine. And it was largely identified in children. So I think Sweden has Sweden is coming to this decision perhaps with some recent experience of potential harm in children from a pandemic vaccine, and also from a situation where our children are in school. So it's not been something, contrast this with, say, my brother in the United States, whose 3 children have been at home for almost a year. And they're looking at vaccination as a way to get teachers and children back to school. So I could see that the considerations in the United States may be different than, say, here in Sweden. But from my understanding, trials in children, I certainly have seen calls for children to be included in particularly the AstraZeneca vaccine trials, and maybe even the Novavax vaccine that's oncoming. But I don't know if Irene has any reflection on that in Israel. What are they doing there, the recommendation?
The children are vaccinated from 16 to 18. They are all vaccinated, not all, but they have started to vaccinate between 16 and 18. And for the time being, it was said that perhaps we will wait for the clinical trials in children. That will be perhaps we will have some results from Pfizer in the August or something like that. But it's absolutely. It was yesterday. It was somebody from the Ministry of Health who was in a conference say that certainly not before the August. We will not have anything about that. So, but children have been locked down for a long time in Israel. I think that we have them longer time in lockdown also in the world. So I think that it was quite a difficult thing. So now we have decided to set up a registry of pregnant women and to send it to the in ISoP Israel. No, not so that we will be in contact with, of course, with the Ministry of Health, and I suppose that Ministry of Health is in contact with UMC, so you will have the cases.
Your personal point of view and the point of view of the WHO or the Uppsala about the need of this. Not only pregnancy registry,
Yeah.
but observational studies that assess both effectiveness and safety.
Yeah. I mean, my personal opinion, I mean, I'll have to be stating for, I mean, UMC doesn't have an official position on this. So of course I'm speaking for myself. But from my understanding, I mean, working with the WHO and knowing what they have made recommendations in their guidance for safety surveillance, I mean, I certainly believe that the recommendation, if at all feasible, is for active surveillance, specifically for safety. But I mean, I'm not as in tune with the position on studies for follow-up of effectiveness. But of course, you can always consider a lack of efficacy or a lack of effectiveness a safety problem, of course. So I don't know that I have enough experience in either effectiveness assessments or even active surveillance myself just to speak anything further, I must say. But I know that it's always considered a benefit if you're able, if you have the resources and the ability to perform active surveillance in addition to passive surveillance.
I think that this is one of the reasons why Israel has received so many doses of vaccine, is because we have 90% of the medical data are digitalized. So, and there is a very strong epidemiologic research on this data. So this database are analyzed on real world data. And I think that in the 2 or 3 coming weeks, we will have the analysis of this real world data. And I think that this is certainly one of the main reasons why Israel has been chosen to have all this massive campaign, because we are able to produce this real world data on safety and efficacy.
There's one other question about the anaphylaxis, and that it's often confused with uncomplicated syncope by the reporter, and if there are any recommendations could be made to improving the reporting of ADRs such as anaphylaxis. You know, I don't know. When I get a question like this, I'll have to admit I'm usually in the seat. I have to admit that even though I'm a physician, I never reported an adverse event myself. And so my time in pharmacovigilance has always been on the side of the receiving of the reports. And so most of my recommendations and my perspective on this comes from the other side. So I think it's my duty and the duty of other people in pharmacovigilance to educate those who receive the reports, as well as people who make the reports, on the importance of capturing the highest quality information that we can get. I know that it's very difficult in reality sometimes to provide a high quality report, but certainly when I do my teaching from the UMC, that we encourage those working in national centers that when they have a report of an adverse event of special interest, particularly anaphylaxis, to use Brighton collaboration case definitions as a resource to collect additional information.